Look at types of special senses. So the special sensory receptors are for touch, taste, smell, sight, hearing, and equilibrium. We're going to talk about special senses that are able to process chemical, mechanical, and electromagnetic stimuli and turn that into an action potential and then convey that sensory action potential to the central nervous system. The eye and vision is what we'll start with first. 70% of all sensory receptors are in the eye, and nearly half of the cerebral cortex is involved in processing visual information. Most of the eye is going to be protected, and it's mainly going to be protected in its side or posterior areas by a cushion of fat and the bony orbit. Some associated structures are the eyebrows, the eyelids, eyelashes, conjunctiva, lacrimal apparatus, and extrinsic eye muscles. The extrinsic eye muscles move and maintain eye shape. The lacrimal apparatus is your tear duct. Your conjunctiva is a mucous membrane that covers the eye anteriorly and also lines the inside of your eyelids. Your eyelashes initiate reflex blinking. Your eyelids protect the anterior part of your eye, and they are also going to be able to secrete a whitish oily secretion called Sandman's eye sand. And then you have your eyebrows, which are superior to the eye itself and provide shading and prevent perspiration from reaching the eye, so they're protective. Here are those structures. We have the eyebrow and the eyelids. You have an upper and lower eyelid. They're also called the palpebrae. The conjunctiva is hard to see here, but it's covering the white of your eye and is merging with the cornea. We have the palpebral fissure, which is the space between your eyelids, a lateral and medial commissure, the points of your eye. The iris is the colored part of your eye, and your pupil is the black part of your eye. We can also see the sclera, but it's under the conjunctiva. That's the white of your eye. And then the lacrimal caruncle is what your tears will drain into to enter the lacrimal sac. Next, we'll look at your eyelids more, or your palpebrae. The tarsal gland is the gland that is located in your eyelids, and it lubricates the eyelids, prevents them from sticking together. And the tarsal plate is connective tissue that helps support your eyelids. Tears contain a lot of chemicals, and they're able to kind of digest or trap or fight pathogens if they come across them as they sweep lateral to medial in your eye. They're going to enter the eye via some superlateral excretory ducts from the lacrimal gland, and then they're able to exit the eye medially via the lacrimal punctum. So they drain into the lacrimal sac, and then that becomes the nasolacrimal duct, and then they kind of exit out your nose. So here is the lacrimal gland, and it has several excretory ducts. So tears would drain from the gland via the excretory ducts across the eye, enter the lacrimal punctum, then drain through the lacrimal canals into the lacrimal sac, then down and into the nose via the nasolacrimal duct. Now you can experience watery eyes if any of this gets stopped up and prevents drainage via the lacrimal punctum of tears. Next, we're going to look at 6 extrinsic eye muscles. They are strap-like. They're outside the eye. They're able to move the eye and maintain its shape. There are 3 cranial nerves that innervate the extrinsic eye muscles: the oculomotor, trochlear, and abducens nerve. 4 of the 6 extrinsic eye muscles are innervated by the oculomotor nerve, and the lateral rectus is innervated by the abducens nerve, and the superior oblique is innervated by the trochlear nerve. All of the rectus muscles are at least telling you part of what they do. The lateral and medial rectus are the most obvious. The lateral rectus moves the eye laterally. The medial rectus moves the eye medially. And then anything that's superior or inferior has 2 movements. If it's a rectus muscle, we're still at least getting some information from the name, because the superior rectus elevates the eye and the inferior rectus depresses the eye. And then their second movement for both superior and inferior rectus are medially. So it'll elevate and move the eye medially, or depress and move the eye medially, depending if you're using superior or inferior rectus, respectively. The oblique muscles are kind of almost telling you a lie. They're going to do the opposite of what their name suggests. For example, the inferior oblique actually elevates the eye. The superior oblique depresses the eye. So inferior suggests movement downwards or towards the feet, but it's actually going to move the eye up. and then same thing for superior oblique. So that would suggest moving the eye towards the head or upwards, but it actually moves the eye down. And then for the oblique muscles, the second movement is lateral. Next, we're going to take a closer look at the structure of the eyeball. The eyeball is going to be mostly spherical, but it's a little irregular. It's hollow. It contains some fluids. We will separate it into an anterior and posterior chamber, but it also has an anterior and posterior pole. You'd find on the anterior pole the cornea, or it could be covered by conjunctiva, and the posterior pole is going to be made up of the optic nerve. The lens is going to separate the two cavities, the anterior and posterior segments or chambers, and we see that the lens is held in place by suspensory ligaments, which are attached to a ciliary body or a piece of muscle. There are 4 structures we want to point out in the posterior compartment, and that would be the fluid first, that is in the posterior segment, and the fluid in the posterior segment, so behind the lens, so we're looking at this part. The fluid in this segment is jelly-like and called the vitreous humor. And it's going to make sure that the retina, which is here in yellow, the retina stays pushed up to the back of the eye, so it maintains intraocular pressure. The retina is the innermost sensory layer of the posterior compartment. Next, we'll look at the choroid. The choroid is the middle vascular layer of the posterior compartment. And then last but not least, we'll look at the sclera. The sclera is also known as the white of your eye, and it's going to be the outer fibrous tunic of the posterior compartment. So in the posterior chamber or compartment is the vitreous humor, holding the retina in place. The retina is the inner sensory tunic. Then we have the choroid, the middle vascular tunic. Then there is the sclera, which is the outer fibrous tunic. So we'll talk a little bit more about those, but you can also see that part of the retina exits the back of the eye. The area where it's exiting the back of the eye, the yellow, that is the optic disc. The structure that is actually exiting the back of the eye, that is the optic nerve. The optic disc is also known as your blind spot. It's an area where we have no photoreceptors, no visual processing, and we have to thank our cerebral cortex for filling in that information, so we don't have a true blind spot in our vision. On the front, we just want to acknowledge the lens. So that's separating anterior from posterior chamber or compartment. Point out our suspensory ligaments, holding the lens in place, and the ciliary body attached to the other end of the suspensory ligaments. There's also the cornea right here, most anterior, and separating the cornea from the lens is a fluid called aqueous humor. So aqueous humor is in the anterior chamber, vitreous humor is in the posterior chamber. Then we also have some more muscle, which is your iris. And then the space, so this is your iris, the muscle or material that surrounds the space called your pupil. And we'll look at all these structures in more detail. First and foremost, your lens. It's biconvex, it's transparent. It's important that it's transparent. That's why it's also avascular. We want to make sure that light can go through the lens without being obstructed. It does allow precise focusing of light into the retina, so light would pass through the cornea, aqueous humor, through the pupil, and then through the lens. And then it would travel through the entire posterior chamber to the back of the eye and hit the retina. The lens does age, and it kind of changes with age. It becomes a little more dense, more compact, and doesn't change shape as well, so it loses its elasticity. Next, we're going to move and look at the tunic, so the posterior chamber, and we'll look at the retina first. The retina is the sensory tunic because it contains photoreceptors, but the sensory tunic itself is made up of 2 layers, the pigmented layer and the neural layer. The neural layer is actually the innermost layer and has in it many photoreceptors. And these photoreceptors are able to turn light energy into chemical energy. Then there's the pigmented layer, which is behind the neural layer but catches the light and then conveys it forward to the neural layer, and then it's going to help convey that information to the ganglion cell axons, which become the optic nerve. So the pathway of light traveling through the eye is it passes through the cornea, through the aqueous space, then through the pupil where it hits the lens. As it leaves the lens, it travels through the posterior chamber and hits the retina. So let's move down here to the other image. When it hits the retina, it's actually traveling to the pigmented layer, and it's going to be processed from this pigmented layer backwards, picked up by 1, 2, 3 neurons, which makes sense because there is a 3-neuron path at the circuit level for sensory processing. The first neuron in the visual pathway is the photoreceptor. Then the signal is going to go to a bipolar cell. Next, the signal is going to go to a ganglion cell. It's the axons of the ganglion cells that you see here in yellow. They make kind of a 90-degree right turn and then another one. And all the axons of the ganglion cells become the optic nerve. So the photoreceptors are able to transduce the light energy into chemical energy or an action potential, then convey that to the bipolar cells, which convey that action potential then to the ganglion cells. And it's the ganglion cells, again, forming the optic nerve. The retina itself is really delicate and fragile. It really does depend on the vitreous humor to push it and hold it in place against the back of the eye. We've talked about that area, the optic disc, the blind spot where there's no photoreceptors. And this is because it's the ganglion axons that are leaving the eye in this location. So there's just not room for photoreceptors. But there's plenty of photoreceptors in the retina. There's rods and cones. So let's just point out the structures again that we're about to talk about. So here again, the back is the pigmented layer. In the front, so this whole segment, I'll color in part of it. This part is the neural layer. And light would come back and hit the pigmented layer first and then travel forward. So it'll be picked up first by a photoreceptor, then a bipolar neuron, then a ganglion neuron. And the axon of the ganglion neuron takes some right turns, and a bunch of those axons become the optic nerve. So the photoreceptors themselves could be rods or cones. The rods respond to dim light, the cones respond to bright light. The cones pick up high acuity color. The rods pick up kind of gray or dark tones, and so they're used for your peripheral vision, where cones, again, are used for high acuity color vision. And they're being named based on their structure. So a cone has a cone head, and a rod has a rod-shaped head. So not very complicated as far as what their name is coming from. But make sure you know cones, color, and they pick up color best in bright light. Rods, gray tones, and they're used for peripheral vision. So that's the basics for your photoreceptors. And also notice that our photoreceptors are near that pigmented layer. So they're really able to pick up that light that was embedded in the pigmented layer, waiting for processing. They're able to pick up that kind of wave-like light energy, and then they're going to be able to convert that to chemical energy. We can also see the pathway of light a little better. So it would go photoreceptor, bipolar neuron, then ganglion neuron, and we can see all these axons taking a right turn, and they eventually become that optic nerve. So some functional characteristics of rods: very sensitive in dim light. We got more of them than we do cones. They're great for night vision, peripheral vision. Again, their input is kind of dark or gray tones. And another thing is that their pathways converge. And what that means is they're going to send multiple signals to one bipolar cell, and that's going to result in a fuzzy or indistinct image if we're using it for night vision or peripheral vision. That's why us humans, we don't have really great night vision, because our pathways converge when we use our rods. So let's look at an example here. I have 1, 2, 3 rods, and they're all sending their signals to this one bipolar neuron. So their signals are all converging to that one bipolar neuron. That's a lot of signal to take in for one cell, and it results in that kind of fuzzy imagery. Now for cones, we use them for high acuity color vision. So we have non-converging pathways. Every cone gets its very own bipolar cell. So we get really high acuity, kind of pinpoint, great color vision. But they need bright light for that. So they need bright light for their activation. So they have low sensitivity. You got to give them a lot of light for them to say, oh, I recognize that there's things to see. And they provide high resolution color vision. Next, let's talk more about the chambers. The chambers of the eye are a front and a back one. So the front chamber is an anterior chamber. The back chamber is a posterior chamber, sometimes called areas or segments. The posterior segment contains vitreous humor, that's the jelly-like type humor, or fluid that's in the back of the eye. It's able to transmit eye through the posterior compartment. It's going to support the back of the lens, but again, it's really helping to hold that neural layer of the retina firmly against the back of the eye, against that pigmented layer. So that intraocular pressure is paramount for holding that part of the sensory layer in place. And a good thing about vitreous humor is it forms an embryo and lasts your entire life. In the anterior segment, we have aqueous humor. Aqueous, like watery. It's not as jelly-like. It's not as thick. Kind of like blood plasma. So it's a similar kind of fluid consistency. But also like blood plasma, it's continuously formed. And it drains, and then after it drains, we make more. So it supplies nutrients and oxygen mainly to the lens and cornea, but also to the retina and removes waste. Now, if we block the drainage of aqueous humor, a condition called glaucoma can develop. And this is just basically where the aqueous humor kind of drains into the back or posterior compartment and puts a lot of pressure on that retina. So increasing intraocular pressure can be bad because if we put pressure on the retina, especially near that optic nerve, it can result in blindness. So let's just point out the areas where the fluids would be. So here is our anterior chamber. Inside that would be aqueous humor. Here is our posterior chamber. We actually see our vitreous humor sitting in the back. And then we can also see our optic nerve. We have our 3 layers: sensory, vascular, and fibrous, or retina, choroid, sclera. Those are the posterior tunics. There's counterparts to the posterior tunics in the front of the eye. For example, you can see that the choroid is also going to include the ciliary body and the iris, and that the sclera in the front becomes the cornea. So I say posterior tunics, but they do have counterparts in the front of the eye. But you see them mainly as layers in the back of the eye. Let's look at the lens a little bit more. We know it's avascular, it's biconvex, it's transparent. We talked a little bit about it being flexible, but it can change shape. And it changes shape to focus light on the retina. That's where all the photoreceptors are, so we really want to focus light on the retina. The lens is also enclosed in a thin elastic capsule. It's held in place by those suspensory ligaments of the ciliary zonule. And we want it also to remain clear, remain transparent. So we want to prevent a cataract or treat cataracts. And what a cataract is a clouding of the lens that causes the world to appear distorted, and it could look like you're looking through frosted glass. Now, it can be congenital. Someone can be born with it, but a lot of times it's age-related or it's as a complication of another disorder, such as diabetes mellitus. Now, surgeries can remove them, but cataracts can come back. But again, we're really looking at the fact that the lens can change shape and other things in the eye can change shape. One thing you might not think about changing shape is your pupil. Your pupil regulates the amount of light entering the eye. And so depending on what you're looking at, will either constrict or dilate our pupil. So pupil is regulating the amount of light. So if we make the pupil smaller in diameter, less light can get into the eye. If we make the pupil larger in diameter, more light can get into the eye. So for far vision, so if we're looking at things that are distant, we actually want to dilate our pupil. we'll have a pupil dilate, more light can enter our eye. Hopefully more information, therefore, can be discerned about this thing that's far away from us. For close vision, the thing's already really close. We can see it pretty well. We don't need a lot of extra light entering our eye. It might actually make the image fuzzy. Now, with distant vision, we can't really see the objects. We want as much information as possible. It doesn't matter if it's fuzzy. With close vision, we want less light entering the eye, so the pupil will constrict. So with close vision and bright light, pupils constrict. With distant vision and dim light, the pupils dilate. And changes in emotional state can also cause the pupils to dilate or constrict. They'll definitely dilate when something you're looking at is appealing or requires problem-solving skills. All right, next we're going to talk about the light that we can convert into chemical energy. So light is what our eyes respond to. The visible light that goes through your eye hits the pigmented layer of the retina. But what we see is really only a small portion of the electromagnetic spectrum. So there's a lot more kind of light waves that could be perceived, but our eyes can't pick up on them. Keep in mind, we're not going to go into a physics or quantum mechanics explanation here, but lights travel both as waves and as packets of energy called photons. So they have like a dual nature. So just know that be aware they have a dual nature. Light's more complicated than we want to really get into. But rods and cones are able to respond to different wavelengths and amounts of photons of the visible spectrum. So let's kind of start from point A and get to point B, or get to our retina where light's going to be focused. As light passes from air into the eye, it's going to move in this order. So first it moves through the cornea, then it moves through the aqueous humor of the anterior chamber, then it's going to pass through the pupil between the iris as it enters the lens. So it's going to travel through the lens, it'll enter and exit. It'll exit the lens and enter the vitreous humor of the posterior chamber. Then it'll hit the pigmented layer and finally be processed by the entire neural layer of the retina. The first part of the neural layer of the retina is the photoreceptors. So that processing will require 3 neurons: photoreceptor, ganglion cell, and bipolar cell. So first it will be processed by the photoreceptor, then the bipolar cell, then the ganglion cell. And throughout this focusing, light's going to be bent 3 times when it enters the cornea and when it enters and exits the lens. So what we actually see, or what hits our retina, is upside down and reversed right to left. The only reason we don't view the world that way is because our cerebral cortex fixes that for us. In the integration or processing step, that's fixed. A couple of other things happen besides kind of our pupil changing shape if we're trying to focus for distant vision. Our eyes are actually best adapted for distant vision. So other than our pupil dilating, light rays that enter the eye from distant objects don't really need to be refracted a lot and are able to hit the retina. So in a case like looking at something that's further than 20 feet away, we would see that the ciliary muscles are relaxed and the suspensory ligaments are going to be held tight. And that means that the lens will flatten. So for distant vision, we're going to need a flatter lens, and we get a flatter lens if we tighten the ciliary zonule, which is the suspensory ligaments, and we can do that by relaxing ciliary muscle. Now this is controlled by sympathetic activation. Sympathetic activation will cause relaxation of the ciliary muscle. So again, ciliary muscles are relaxed, the lens is stretched flat by the tension in the ciliary zonules, which will be tight. This occurs if we're looking at something that's further than 20 feet away. Something further than 20 feet away is considered far for a normal or emmetropic eye. A lot more has to happen for close vision. These 3 things have to happen: accommodation, constriction, and convergence. Convergence is just where your eyes are going to immediately rotate kind of towards each other in order to look at the object being viewed up close. Constriction is where we're going to see, again, something's up close, so our pupil is going to constrict. This is a pupillary reflex, and it's really just supposed to prevent divergent light rays from entering the eye. And then accommodation has to do with the lens changing shape. No accommodation was needed for distant vision. That's kind of the normal state of our eye. For accommodation, we're going to see the bulging of the lens. It's not going to be as flat. We're going to see the ciliary zonule or the suspensory ligaments be loose. And we're going to see that the ciliary body, which is muscle, is going to be contracted. So for focusing for close vision, the ciliary body contracts, which loosens the ciliary zonule or suspensory ligaments, and that allows the lens to bulge. Constriction occurs, so the pupil constricts reflexively, and that reflexive constriction is going to prevent divergent light rays from entering the eye, and our eyes are going to immediately rotate in order to look at the object being viewed. Now, one homeostatic imbalance we want to talk about is presbyopia, which is the loss of the ability to accommodate over the age 50, which will make close vision a lot harder if you're over that age. Depth perception is only achieved with both eyes. So if you think you have depth perception using one eye, that's only because of learned clues from kind of your life experiences that you're using to determine depth. But true three-dimensional vision requires not only both eyes, but fusion of that information to certain parts of the cerebral cortex. Next, we're going to look at some developmental aspects of the eyes. And first and foremost, vision is not fully functional at birth. Babies are hyperopic. They can't see up close as much, so they're going to be farsighted. They see only gray tones, so their cones aren't working as well, and their eye movement is going to be very much uncoordinated. But that'll get better as they age. Depth perception, color vision, usually that's going to develop about age 5. And then with age, the lens is going to lose clarity, so it can become cloudy, cataracts become more common. The dilator muscles of the iris may become less efficient, so it'll be harder to constrict or dilate your pupil reflexively. And then visual acuity is actually going to drastically decrease by age 70. Problems with refraction you're probably a little more familiar with are myopia, hyperopia, and astigmatism. Now, we talked about hyperopia. Yeah, that was a tongue twister. Hyperopia occurring in babies, but it can also persist. And again, that's farsightedness. This is when the focal point is behind the retina. In myopia or nearsightedness, when the focal point is in front of the retina. But the takeaway here is that with both nearsightedness and farsightedness, the light is not hitting right on the retina as it's refracted when it goes through the cornea, enters and exits the lens. And we'll look at that more in just a second. But an astigmatism is also another problem with refraction, and it's usually caused by unequal curvatures of different parts of the eye, mainly the cornea or the lens. But we can correct all of these problems. For example, nearsightedness is corrected with a concave lens. Farsightedness is corrected with a convex lens, and we can actually ground down your lens or your cornea or put in implants. And now there's even laser procedures in order to fix an astigmatism. So here is a normal, what's called an emmetropic eye. And we can see that light enters the cornea, the lens, and exits the lens. And then it'll be refracted and all pointed to a certain focal point, hopefully on the retina. That's what happens in a normal eye. But with a myopic or nearsighted person, all of that refraction leads to the focal point being in front of the retina. With hyperopic eyes or farsightedness, it leads to the focal point being behind the retina. And so that's not making it easy for the photoreceptors to catch or conduct this light towards the axons of the ganglion cells. So we can fix this. We're just going to add another layer of refraction. So for the correction on a nearsighted person, we'll give them a concave lens. For a farsighted person, we'll give them a convex lens. And that will add another layer of refraction that can correct these different kind of problems in refraction that we call farsightedness and nearsightedness. And next time, we're actually going to look at the chemistry of visual pigments using rhodopsin and rods as our example. So stay tuned for that. Make sure you review the different parts of the eye.